All right, let's get right into it. We're going to tackle one of the biggest questions in neuroscience, resilience. I mean, what makes the human brain so good at bouncing back from stress? And what's happening when it doesn't? Well, a really fascinating new study gives us a whole new way to look at this. And believe it or not, it might all come down to a single overlooked protein. This is a question that I think we can all connect with on some level. We all face stress, right? But our responses can be completely different. For some people, a tough time is just a temporary setback. But for others, it can be the trigger for something much more serious, like major depression. And this difference, it isn't about willpower. It's about biology. And scientists are finally starting to crack the code. And I want to be clear, this has been one of the toughest nuts to crack in brain science. For decades, researchers have looked at everything: genetics, our environment, different brain circuits, you name it. But a really clear molecular answer for what makes a brain resilient versus what makes it susceptible, that's been tough to find. That is, until now. Because a study that just came out in Science Advances has pointed to a brand new player in the story, a specific protein that seems to act like a master switch for resilience. So, let's follow the clues just like the scientists did. So our story starts with the very first clue. The researchers found something was consistently missing in the brains of people who had depression. So this first big hint actually came from looking directly at human brains. Scientists were examining the hippocampus. That's a key brain area for mood and memory. And they found that in people who had been diagnosed with major depression, the genetic blueprint for making a certain protein was way down compared to people without depression. This was the very first sign that this protein might be important. And that protein is called Zkscan4. Now, you're going to want to remember that name because it is our prime suspect. The data showed this really clear pattern. Less Zkscan4 in the hippocampus seemed to be connected with depression in humans. And what's really interesting is that this wasn't just true in humans. The scientists saw the exact same pattern in mice. After they exposed mice to stress, some of them bounced right back. They were resilient. But others became what they call susceptible, showing behaviors that look a lot like depression. And when they looked at their brains, you guessed it, the susceptible mice had way lower levels of Zkscan4, perfectly mirroring what they saw in the human patients. So this leads to the million-dollar question in science, right? Is this low level of Zkscan4 just a side effect of depression, or could it actually be causing the vulnerability to stress in the first place? To figure that out, you can't just observe, you have to run an experiment. And that brings us to the moment of truth, the crucial test of their whole idea. The researchers designed this really elegant experiment to see if this protein really is the key to resilience. To do this, they used a really powerful tool called a knockout mouse, and it is pretty much exactly what it sounds like. These are mice that have been genetically engineered so their brains just cannot produce the Zkscan4 protein at all. They are completely missing our protein of interest right from birth. Okay, so here's the setup. First, you've got these mice that have no Zkscan4. Second, and this is the really clever part, you apply a very mild stress, something that normally wouldn't even faze a typical mouse. And then you just sit back and watch what happens. The results were, well, they were striking. A normal mouse, when it's faced with this tiny bit of stress, it just acts normally. It's social, it explores its space. But the mouse without Zkscan4, it reacted completely differently. It became withdrawn. It avoided other mice. Just the absence of this single protein seemed to make it vulnerable. So this is the critical point. Just removing that one protein was enough to turn a resilient mouse into a susceptible one. See, this isn't just a correlation anymore. This is evidence of causation. It really looks like Zscan4 is essential for resilience to stress. But how? How does it do it? Okay, this is where the story gets even cooler, because it turns out Zkscan4 doesn't work in isolation. It's actually connected to one of the most famous systems in all of brain science, the serotonin system. Probably the best way to think about what Zkscan4 does is to picture it as a kind of molecular brake. Its whole job is to slow things down, to keep certain processes from going into overdrive. Technically, it's a transcriptional repressor, which just means it stops certain genes from getting turned on too much. And what specific thing is it putting the brakes on? A very, very important one, the gene for the serotonin 2A receptor, also known as HTR2A. Now, this receptor is a huge player in our mood, and it's also the main target for psychedelic drugs. So Zkscan4's job is to keep this powerful receptor from getting out of control. So it creates this really simple system. In a resilient brain, where there's plenty of Zscan4, the brake is on. This keeps that HTR2A receptor in check, and brain signaling stays nice and balanced. But when Zscan4 is missing, that brake is off. The HTR2A receptor goes into overdrive, and that disrupts the brain's circuits, creating vulnerability to stress. And here's the proof. This chart shows the actual protein levels of that HTR2A receptor in the brains of the mice. And it's just what the brake analogy would predict. The mice without Zkscan4 have significantly higher levels of the serotonin receptor. The brake is truly measurably off. Now, this incredibly detailed molecular picture isn't just amazing science. It actually points directly to a potential new way to think about treating depression. This leads to the ultimate test, right? If the whole problem is an overactive serotonin receptor because of a missing brake, then what happens if we manually fix that receptor? Could we actually rescue these vulnerable mice and make them resilient again? So the scientists tried two different ways to fix the problem. First, they used a drug to block that overactive HTR2A receptor, essentially putting on an artificial brake. And second, they tried reintroducing just a small piece of the missing Zkscan4 protein directly into the brain to see if it could restore the natural brake. And the results were a huge success. This chart here shows this social behavior of the mice. That low bar on the left, that's the vulnerable knockout mice, avoiding social interaction. But look at the next two bars. Whether the scientists blocked the overactive receptor with a drug or added back a piece of the Zkscan4 protein, the result was the same. The mice went back to normal, resilient social behavior. They were rescued. The authors of the study really sum it up perfectly themselves. They write, "Our findings demonstrate an essential role of Zkscan4 in promoting stress resilience, suggesting a potential antidepressant effect of Zkscan4 133, which is really highlighting the potential of using a piece of the protein itself as a treatment." And that leaves us with this really provocative final thought. For decades, our main antidepressants have worked by just flooding the brain with serotonin. But what if that's too blunt of an instrument? This research suggests a much more precise approach. What if the future of treatment isn't about more serotonin, but about restoring the brain's own natural braking system simply by boosting this one critical protein? It's a profound shift in thinking, and it all started with the simple clue of a missing protein.